Should I move on to a couple announcements while people finish introducing themselves in the chat?
Yeah.
Awesome. All right. Welcome everyone to the Lattice Lightsheet User Group, hosted by Brendan and Claire. And we'll just have a couple of announcements and the introductions in the chat that we've been doing. And then we'll have a presentation from Andrew from 3i in a little bit and wrap up. And our next meeting will be April 2nd. So Claire and Brandon are our wonderful hosts, and I'm Vanessa. I'm the BINA Program Administrator. I help coordinate these meetings along with my colleague Nikki Biley, who's our BINA Program Coordinator. This is the introduction slide. We've been over this one. Continue filling out your favorite color of fluorescent protein and your major challenges that you're facing right now. Upcoming BINA activities, we have our scientific speaker series is celebrating Black History Month, and we have a speaker every Tuesday at noon Central. So we've got a speaker today kicking it off. Dr. Bell will be presenting, and then later in the month we will have Brian Millis presenting about his career, and that series will continue on the third Wednesday of every month. So please join us for that. Here's just a layout of all the activities going on in February to give a kind of calendar view of it all. So we hope you can join us for many of those. We also have our exchange of experience virtual groups. I believe the next one is going to focus on building and setting up a new facility and kind of what considerations you might want to consider when doing so. Just to share that we have a new flyer about BINA. It's available on our resources page, which is also a new page. Hopefully you've seen about that in the newsletter. If you're not a member, please join us. But we just wanted to share this is available and thank Jason, our member of our communications and early career working group for providing that. And of course, we also want to thank our platinum sponsors. This is a new thing we're doing this year, but we wanted to give recognition to the companies that are really strongly supporting us. So AVR Optics and Miltenyi Biotech, thank you for your support. And with that, I'll pass it over to Brandon to do some introductions, announcements, and etc.
Perfect. Yeah, so Andrew, if you wanted to start sharing your screen. So I was just looking, Andrew, it's been at 3i for quite a while now. It looks like going on 8 years as the product manager there. I'm super excited to hear about what you guys have done with the Lattice and what's maybe new. And I think you have a demo for us as well. Is that correct, Andrew?
That is correct. Yes. Oh. Don't judge my my poor cell biology skills today, but
judging so hard. So perfect. Andrew, you want to take it away? I think you have full screen share.
I will, yeah. I'm going to say hi before I screen share. So,
of course, yes, please.
Thanks for the lovely introduction. You're right. I've been here for almost 8 years now. My background is technically a pharmacology PhD, but basically cell biology, all around microscopy, but intravital. So I swung from mouse work over to look inside cells of the lattice, which is what we're going to talk about today. So I'm going to share this PowerPoint, and hopefully all of you can see this. What I'm going to focus on today is kind of what we have done with the Lightsheet project, and how long we've been working on it, changes we've done in comparison to something you might have seen from Zeiss. I've presented on theirs here as well as differences kind of with the home-built systems that have existed for a while in the community. So next, just in case people don't know who we are, I'm assuming most of you do by now, but I'll start with a really quick introduction of us as a company. Go over, like I said, key features about the Lattice, answer some questions, and then give you a relatively brief but live tour of the system I have sitting next to me here in our office in Denver.
Andrew, just a quick question.
Yeah.
Your audio is very crackly. I don't know if there's anything you can do on your end. If not,
Yeah,
it's audible, but it's it has a bit of distortion or something in it.
Is this better?
Much better.
Okay, cool. I think it was just a webcam issue.
Perfect. Thanks.
Thank you for letting me know. I can't hear myself, but I get this. That's nice. So as I mentioned, we as a company, 3i, have been around for a while now, over 28 years. Started in the mid-90s, built around our software package, Slimebook, that you'll see me use later. The main innovation at the time was deconvolution on a computer, back when that was cutting edge technology many years ago. Obviously, we still do that, but we've branched out into hardware integration and hardware manufacturing. So rough timeline that you can see here. Obviously, I'm highlighting the Lattice today. That was introduced to our portfolio back in 2015 in a partnership with Zeiss Genelia and Eric Benson Consultant. And as we've moved on, we've innovated in other light sheets, other techniques that you can see there. Feel free to ask me about any of those after the fact, but obviously we'll focus on the Lattice here today. The other thing that is relevant is we have a lot of light sheets. So again, focus today is the Lattice. But when you think about light sheets and you think about what you want to image, which I'm sure many of you already have, the Lattice is what you're thinking about when you're imaging cells, intracellular events, maybe cell-cell interactions, object portions of larger things. And then we have two other products and systems in the kind of mid-sample size embryos, zebrafish, things like that. And then the larger cleared regime with our brand new Axle 3-tissue light sheet that we just launched at the end of last year.
So Andrew, there's still a few people saying they can't hear you now. I think it's more just quieter. I don't know if you can get closer to the microphone or speak a little bit louder.
Is this better again?
Yes.
Is it crackly?
No, it's good.
Cool. I don't know what Zoom is doing with the microphone. My apologies, everyone. This isn't the first time we've done this, so should all be working fine. Well, if you can't hear again, just interrupt me, please. I'll also speak louder and scoot closer. So this slide here kind of highlights the very broad overview of what we've done as an evolution of the Lattice light sheet since that has been in our portfolio. So at the beginning of the projects, we were producing and installing essentially direct clones of what was at Genelia at the time. So I kind of have that represented by year 0 here in the top left. This was only slightly tweaked just because of some differences in parts that we could obtain from what Genelia had, but basically the same thing, where you had water heating for the environmental bath, a manual mask, the same light path, older, but at the time, current cameras, a pretty free-beamed laser launch. So we, as part of our agreement, started installing those and learning ourselves. And we made a commitment to make pretty significant changes to make it both easier to use and more optically stable. And so in year 2 and year 3, we were kind of iterating on that. Year 3 was what we called our version 2 that we released, where we had motorized most everything that you would want to touch, making sample loading and changing the mask a lot easier. Also just improving the laser safety around it, improving the beam shielding in general, making it optically more stable. We add a custom breadboard that was different from the initial one, actually made for us by the same people who make our Airtables, TMC. And this was great. This really was a nice improvement to everyone who had used the original system, probably similar to anyone who has used a home-built system. Many people here who have also used one of ours in the past. Well, that wasn't quite where we wanted to stop, right? We want to continue to innovate, especially as our friends at Zeiss have released their version of the system. And we've continued to modify it into what you see here in the today photo. And that's actually the same system I have sitting next to me, where we've shrunk it down even more and added a lot more ease of use features, including some software and some laser tweaks, just to make it in general better for the general user. So the highlights that I want to go over today, kind of as a slightly differentiating ourselves from maybe some of the stuff on the home built side, but also some of the stuff on the Zeiss side. I'm going to start with structured elimination. So this is something that our system is completely capable of doing, both on the acquisition and the analysis side of things, where you can acquire super res images just using the inherent lattice pattern. And I'll talk more about that in a couple slides. One of the other additions we've made over the years is we added photo manipulation via our device called the Vector 2. It couples into the beam path through the detection side. And this is a device we've used on our inverted systems and upright, say, spinning discs or wide fields for over a decade. And it made sense just to make easy raster-based ROI photo bleaching and FRAP studies on the Lattice as well. We've kind of, there's 2 options for lasers that I'll talk more about, but we do still give users the options for really, really, really high powered lasers. I mean, 2000 milliwatts is excessive for most users. But if you really want to push the capabilities of the system with some single molecule imaging, that has been useful for some of our groups. We kept a lot of the flexibility in the light sheets, including some new stuff that has recently come out of Betsing's group. We also kept the upright objective orientation. I have a cool case study for why that's still really useful, even though it is different to a lot of systems that are out there. And then full incubation system through our friends at Ogo Lab. So I'm going to start with the sim component of the system. So one thing that a lot of people don't inherently think about when they use a lattice is that because of this way the pattern is structured, as many of you have seen, that multi-vessel side lobes and main pattern actually cause striping across the image. Usually we dither that and shake it so that you don't see the striping on your standard imaging. But if you want to step that using an VX Galvo in a really defined pattern, depending on wavelength and mask, you can acquire 3, 5, or 7 images for every super resolved plane and make a pretty big increase in the resolution of the system, about 1.5-fold in the X and Z components. And because it's still a lattice, it's still super gentle, so it can still go relatively fast and not bleach your samples, making it great for gentle live cell sim. This image here on the left is H2B, GFP image with one of our groups, where they wanted to look at the different packing densities of chromatin. And they were coupling this with some transcriptional factor imaging, where they wanted to see and correlate the pockets of binding of that transcription factor correlated back to how dense the DNA was packed. The standard resolution of the system was not quite good enough to see the differences in chromatin density, but sim was. So they were able to just switch over to the sim mode, same software package, and acquire nice, beautiful time-lapse videos of the nucleus. As a canonical example of what this actually looks like as a comparison, we have just LifeAct in a fixed cell here that I imaged years ago on the system. On the left, standard lattice, not deconvolved, just standard. And on the right, the exact same cell, image in the sim pattern, the sim mode. And you can see the significant improvement in all the little fine details, specifically around the middle of the cell and kind of the leading edge, that are present, especially if you were to do something like deconvolution on the standard imaging, but not quite as apparent. And then I didn't rotate this, but in Z, you get the same kind of benefit that you get in this sim image. And I kept this picture of what the pattern looks like on the right, just to remind everyone that that is the grading that we're using. We're not introducing another optical grading into the system. We are just using the pattern itself for the actual stepping of the pattern. So another thing I mentioned we added was photo manipulation. So the bottom right there is a picture of what it actually looks like mounted on the system. It's a unit called Vector 2, where it has 2 galvos that you can draw an ROI on your sample, script what time point and what laser wavelength you want to use, and then it bleaches and recovers, as you can see in the video on the left here. This was just a membrane dye on some cells we had here, and we just frapped a region and let it recover. So pretty easy and classical frap experiment, but now using the lattice for the actual imaging modality. So again, gentle, fast, nice and easy for this. What we really like about our integration of photo manipulation onto the system is not only is it user-friendly and just ties into our software really nicely, but it couples to the same laser launch that you image with. So we have a fast galvo switcher that is back there that switches between imaging and then the photo manipulation event. So you can use the exact same lasers. You don't have to have a separate laser source. You don't have to buy separate lasers just for photo manipulation. Just increasing the flexibility of what you can do with that same launch. A lot of users maybe have 4 or 5 nanometer lasers that they don't image with a whole lot, just because it's kind of against the idea of gentle imaging. But it's a great laser now for photo manipulation, that if it was in the launch, now it's getting a little bit more use in a device like this for something like photo conversion, where you're specifically hitting with 4 or 5. And like I said, since this goes into the emission path, the field of view of what you're bleaching is already what you're looking at. So there's no complicated math or software tricks that have to be done to transform the beam and what you're actually doing. You just get to bleach at what you're looking at in standard lattice imaging, making it really easy to use. And I wanted to, again, talk a little bit more about the laser options, specifically this exciting black box here on the right called Laser Stack. As you'll see on the system next to me, this is the laser combiner we now install with the system. So the big advantage of using this laser launch is this is the same launch we use on our spinning discs, on our turf systems, on other photo manipulation systems, that we're very familiar with aligning and manufacturing, but it also makes the lattice inherently more stable. When you remove the large high-powered launch that I'm sure many of the home builders here are familiar with, out of the beam path, there's no more open beam path. So there is no more drift that you're correcting for by doing that daily or semi-often beam alignment that you might have to do. Something that we made pretty simple with our version 2 system, but it was still there. Still, if you saw a temperature change in the room, you did have to tweak the beam. That is gone when you couple the fiber optic into the system. Everything holds its alignment as the system is warmed up. And so there's no more adjusting of the beam, allowing us to even more kind of shield off some of the components that you'll see on the system. But due to our familiarity with that other launch, we still offer it as an option, as I mentioned previously, for the groups doing something like single molecule localization microscopy, where they need insanely high power density per voxel to actually do their imaging. So you get to kind of pick. The groups that are doing standard live cell imaging, the laser stack is perfect for them. Even groups doing SIM, the laser stack powers range from 100 to 150, maybe even 200 milliwatts, depending on the laser line, and some brand new that are even 300. So you can get quite a bit of power in this system as well, just not the more exotic powers of the visible wavelengths of half a watt, 1 watt, 2 watt that you might see with the quote unquote high powered launch. And I already mentioned this before, but either of these launches can be coupled with the photo manipulation unit, meaning it has that fast galvo for switching the beam path back and forth so that you can use all the same lasers. A critical component of the lattice itself, obviously, is the annular mask. I'm sure all of you are familiar, especially on the home built side, with using one of these and how it can be tedious to change mask positions on the manual mask of the original design. So one of the things we integrated years ago into our version 2 was updating this to include a motorization of this, and now it's a wheel, to switch between patterns. So you no longer have to look at a back aperture conjugate camera, line up an aperture, move it by hand, and potentially tweak the excitation alignment a little bit to match that better. You can just click a button and it changes to a different mask position. And this lets you have pretty great flexibility in what you can do with samples. So again, the core of this system is going to be live cell imaging, specifically intracellular, subcellular events. But if you have a system and you want to look at something a little bit larger, you of course need to move to a longer light sheet. So no longer, again, moving things by hand, no micrometers are required here. You can just check a different NA from the dropdown and go on an image. And what is great is you don't even need to touch up the alignment. You can look at the beam examining camera, it matches up right away, and it's just really, really user-friendly and easy to use. An example of something big that we've done on here is Arabidopsis, so something big enough that obviously we can see it by eye. So this is a portion of the root, not the actual root tip. This group was looking more at just the tubulin structure in the root adjacent area of Arabidopsis. And you can see as you go deep, you do get some scattering. This is still a plant with quite thick cell walls, but you're still able to penetrate a couple cells down into the edge of the root, something that even on a spinning disc that they were trying to do this with before was pretty difficult. And then, of course, the power that they were trying to image this with on the spinning disc just caused it to bleach. So they were happy to actually get some more gentle imaging of this on the sample, or on the system, sorry. And this was also really easy to mount. I can talk more about that either in the mounting section or during questions later. But the upright objective configuration makes things like this pretty easy as well. So something related to the mask and where we retain some flexibility in writing our own software package for the system is innovations continue to come out of the field, specifically from Betzing and his former colleague, Wes Legant, where they've now published about a year and a half ago for some of this, some novel excitation patterns that they've been working on. I'm sure many of you are familiar with the top left image from below. That is the square pattern or the multi-vessel pattern that most people use, or something similar to that, depending on the NA. It might look a little different. But what the Betzing lab and the Legant lab have begun to work and study a little bit more is by slightly modifying the spacing between the single vessels, you can create other types of patterns. The original Lattice paper talked about hex patterns as well. That's kind of the top right image there, where you're increasing your axial resolution at the cost of also increasing side lobe energy and a little bit of out of focus light. So while this might be great if you are looking at really fine details and need that high axial resolution, it is going to bleach and be a little bit more photo damaging. So, okay, maybe the square pattern might be better for standard imaging. But if you want to kind of combine the highlights of both of those, there's a new class of patterns at the bottom called the hex rect that they named as it's a mix of a hexagon and a rectangle that you can try as well. That gives you some bump in axial resolution compared to the square patterns, but does also get a little bit closer to the hex, and you get a little bit more out of focus light and side lobe energy. And there's a couple other patterns that they published in the paper that we've integrated as well. But Slidebook lets you generate these patterns inside of its interface. You don't have to do this in MATLAB. Not saying that you can't. It just makes it sometimes easier for multi-use facilities. Just have the pattern generator built in. So it's just a drop down that you pick what type of pattern you want. You're selecting the NIA and wavelength based on the lasers you have in the system, and then you can make this these new patterns. Other stuff that isn't even on here yet because it's brand new. There's groups doing kind of the idea of negative light sheet, where you'd be alternating between a square pattern and then kind of the inverse of the square, where it's not exactly like STED, but the idea being that you're suppressing some of the side lobes via that second light sheet and combining those two images after the fact, giving you a great bump in axial resolution, but at an even higher cost when it comes to optical sectioning. So one of our users in India has been working a lot on that and is publishing some really cool data using different patterns like that. Now I want to spend a little bit more time here on sample mounting. Again, all you home builders are used to this, but this is just specifically in contrast to kind of the inverted orientation that Zeiss had talked about recently. And that is that we kept the inherent or the original upright objective configuration of the system. Pictures of objectives that I'm sure you're familiar with here on the left, in sparkling, brand new, clean condition. They usually don't look quite that pretty. But the main reason we did this is this allowed us to keep that really high lateral resolution and not sacrifice that at all. So the 1.1 NA water detection or water dipping lens on the emission side, it's the highest NA water dipper you can get. So yes, it does have the caveat that you have to mount your samples on a 5mm, usually glass, but doesn't have to be glass cover slip between them, just because the working distances are short enough that they almost hit. The trade-off is worth it for most users, where you get that high resolution. The other cool thing about this that I'll talk more about in a couple slides is that cover slip does not have to be glass. I've talked about this in this user group a couple times in the past, but you can make that sample holder out of anything. You could put plastic there. You could put, we've had groups do lithography on that and watch cell migration across a pattern because you're not imaging through it. You're imaging above it. It doesn't really matter what's below. Now, yes, there is a lower objective for a more traditional wide field and kind of sample finding that some people might use, but it's not required, right? If you are able to find your sample with the light sheet, which is usually pretty easy, you can make that opaque and it no longer has to be a nice cover slip. Obviously, again, all of this has to be immersed still in media. So this photo was taken with the front cover removed, but all of that, again, with nice standard kind of flat mounting for most users. This is my very, very basic how you put cells in the samples slides. Again, most of you are probably familiar with this, but the idea being that even though it is still these tiny cover slips, all you do is you seed them into your multi-well dish or whatever cell culture chamber you want to use, and then fix them onto the sample holder. Now, I'll pause here on the sample holder a little bit because this is something that I know we differ from some of the home built community. We have moved away from the little copper clip that holds the cover slips down. Over repeated use, we've just seen them fail, and we're a little bit more worried about users scratching their objectives and then telling us that, hey, you know, your system broke my objective. Can you please fix that for us? So we moved to removing the clip and using vacuum grease just to hold the cover slips down. So that sample holder there in the middle, you'll notice, does not have anything mechanical to hold the clip down. There would just be a little bit of vacuum grease in the inner ring that is cell safe, makes incredibly sticky, that sticks the cover slip down, and then is pretty easy to wipe away with a Kim wipe when you want to just swap samples out. And then again, on the right, you just pop it onto the sample holder into the bath, and we've motorized this, so it's easy to just raise it up again, but image away. Now, a little bit more on kind of a collaboration we've been working with a grad student or PI at the University of Manchester, where she is imaging adult C. elegans and wanted to try the lattice that they had. Well, the problem with that was they had this really special mounting method that involved different adhesive reagents that they had kind of worked out for use on one of their inverted systems. But it was pretty tedious and it didn't give them much control over the mounting. And they didn't really want to use a spinning disc because it was bleaching their samples too much. So Astrid joined us actually in our office here for a little bit. And the two of us worked out these custom 3D printed sample holders for their C. elegans with slightly varying distances in the wells that I'll show in some photos here. But the basic idea is there's grooves lined up vertically on the sample holder. So you can just plop a worm in there and then fix it with the whatever method that you want to use, but that they're always lined up the same way. And that's making them easier to find on something that is a little bit different with that light sheet configuration. So her rough protocol with these 3D printed ones was you still have to paralyze the worms and everything, but you're placing this in this PEG-DA agar kind of this polymer assembly, just so that they stick, and then hold them down with the cover slip so that you can actually make them relatively flat. And if that middle image there is actually the front of one of the sample holders, and if you look closely, you can see those ridges running up and down, spaced apart by maybe about 100 microns or so, that she was attempting to put, a worm in each one of those. They still actually had to get the polymer to cure, so we still put a cover slip on it temporarily on top with some lovely Rain-X to make it pop off easily. But after the imaging was done and it was cured using a mercury lamp, we could then peel the glass off like so. And then you get just the sample holder there that you can mount onto the system. And this was 3D printed, so it was pretty easy to make modifications throughout the project. So as we changed orientations and tried different fiducial markers just to make finding things a little bit easier, or even to correlate the stereoscope work back to the actual sample holder. It took about a week or two, but we were able to get a pretty consistent sample holder set up that we printed a bunch of. I have some sitting in front of me. And then obviously Astrid took a bunch back to use on their system in Manchester. So again, here's kind of a close-up where you can see a little bit more of what the worms actually look like. So if you look closely, the grooves, the arrows have pretty obvious worms in them. We had settled on, for this project itself, wells that were about 75 microns wide, about 150 microns deep. But we had tweaked that pretty easily just with 3D printing being so easy to use and tweak. One of our lovely engineers helped me a lot with this. I'm not a CAD person, but it's pretty easy. And then we even made little spots to make some of the curing stuff easier and adjust the thickness. So you can actually use a little bit of the bottom camera with just shadows if you wanted to. But this was essentially the final result. And where this became really cool was we're actually able to image different motor proteins moving along different neurons in the adult C. elegans. So this is something that they've been able to image before on their spinning disc, but not for very long. They'd usually see a couple movements of these proteins instead of continuous imaging for minutes. And then I don't have the data here for this one because I can't share everything at first. There's some where we did hours. We were just watching these proteins traffic back and forth and looking at different knockouts that she was interested in. But all easy to find and easy to line up, because if you always have the worm in the same orientation and you're imaging it from above with these sample holders, it was great to always say, okay, these neurons will roughly be the same orientation every time. Sure, the worms are going to wiggle a little bit and undulate a little bit, but overall, it was not a guessing game of, okay, let's find the worm and then find the orientation that we want, that I know I've even done with embryos in the system before. So a really cool example of why imaging from above can still be pretty exciting for people. So one of the last things I'll mention before I get on to some questions and showing you the system is environmental controls important. So while we had our friends at Ocolab have made their own. They've also made this, I know, for the home built community. So it's a very similar setup for ours and theirs. It's just a different mechanical structure because our bath is a touch different. But this is an easy way to add digital temperature control, some gas control, not just CO2, but you can do hypoxia and hyperoxia if you wanted to with your gas mixers, and humidity. And on our systems, our solid state heating, we then take off and then add this. I really like their chamber a lot. It's magnetic on the front, if anyone's ever used it. So it actually lets you look at some things without actually removing the sample. It also makes cleaning really easy as well. Because we've been doing this for a while, almost 8 years, same as basically me being at the company, our users have published a lot of stuff now. So we have an example of our publication spotlight there on the right. That's what's on our website. I change it with our web team, every few months or so, where I try to just highlight different papers that our groups are working on. But what I love is it's not all the same stuff. Even though, yeah, sure, this system, great if you're looking at endocytosis or exocytosis. That's not all people do with these systems. So we have groups that are doing single molecule imaging, groups that are looking at T-cell target cell interactions. Our group at St. Jude is doing some of the really exciting SEM correlative imaging, where they're looking at lattice SIM, cryo-SIM, and then actual scanning electron microscopy of the exact same sample, and correlating the movement that they're getting from the lattice to the better resolution that they're getting with those two higher res techniques. There's groups that have done large samples on the system too. Zebrafish, we have a group in Australia that's done a couple zebrafish papers, as well as groups publishing how they deal with data analysis. I know that's been a topic that we've talked about in this user group before. There's still I won't call it an issue, but it's a consideration that Lightsheet user groups have to take. We're sure you're getting amazing data off the system, but what do you do with that? So I'm really happy that some of our groups publish what they do. They publish their cluster computers that they've built and their pipelines that they put onto that. A group in University of Queensland has a great remote access node structure where they can analyze through Slidebook anyone's lattice data offsite. One of the groups, when they published it, he was doing it from a plane, which is a great way of preparing for the conference last minute, as I'm sure many of us do. But even groups that are looking at like the T-cell work and characterizing how you analyze some of these data. And with that, I'll pause on the presentation side of things and take some questions now, and then I can switch over to just kind of showing you a little bit of how Slidebook works in case some of you hadn't seen it before with the Lattice. Thank you as well. I'll look through the chat to see if I see any questions. But feel free to unmute yourself and ask away as well. I am not seeing any questions. Oh, yes. Thank you, Vanessa. I'm happy to share that link. When I'm done with this, I'll either send it to you or just add it to the chat here, but on our website.
It's in there already, I think. And I wondered if you could just give us your email contact information too, so we can pop it in the document that we have. Thanks.
Cool, can do. Yes, Vanessa, that's our general publication link. If you actually go to /Lattice instead of /publications, there's a Lattice specific one. That might be a little bit better for this community. There's my email as well. I believe I'm joined too by a couple of my colleagues, probably here in the chat somewhere too. Do you have more questions? All right, well, I have a question about doing imaging samples covered with a fluorinated polymer film that has the same ND as water. We have tried using FEP before. So, it's a polymer film that has the same RI essentially as water that you can put over the system or over the sample holder and image that way. You have to get it on there, so you still have to stick it down somehow so it doesn't just float away in the bath. But that is something that is possible because it's the same RI. You don't really have to worry about spherical aberration as you're going through the FEP from the sample itself up to the detection lens. So we haven't done a lot of it, but I know it is possible. I've tried it once before with some C. elegans embryos, not the adults, but they were just in kind of a little tube that we then mounted a little bit easier onto the system. thank you for asking that too. Any other? Go on.
Question for you. So beautiful. Have you guys done much with manipulating the sample, like without needing to raise the objectives?
Yes.
So, yeah. You know, near to my heart. Like we're, that's one of the tough things that we always have. Our struggles doing phagocytosis, right? So trying to drop cells on the lattice light sheet, which is
Yeah.
build a new microscope, 'cause it can't do it. So how have you guys solved that? Or do you have
Yes. So I'll say, I can't even say we totally, because a user kind of came up with the idea that I found that works really well.
Sure.
So there is a little bit of a gap above the sample holder, so that allows it to move, right? So the incubation chamber is not sealed.
Yeah.
So what we ended up doing was sticking a like a fine needle right above the cover slip from the side.
Yep.
And then hooking that up to a syringe pump that we could just pump either cells in or liquid in.
Sure.
So that was a great way of adding things right on top of the cover slip. When it comes to media exchange and everything, there's ports in the bath that if you want to do things like that, you can do. And we've had groups add cells that way. So if you wanted to just add a lot of cells, just watch them interact over time, sure, you could technically pipe them in that way too. But doing that little needle right above, you can just fix it to the objective block itself so it stays out of the way.
Yeah.
And since our sample is what's moving up and down for loading, that makes it really easy. The last way that can be nice if you don't really need the fastest time resolution is, since the bath is motorized to load up and down, its position is retained between like clicking load and then going back to the image position. So you could just open the bath, pipette something in by hand, and close it if you want to do that.
Sure.
And obviously that's a little bit more crude, we'll say, and it would have to interrupt imaging, but that does work, and it does return right back to where it was, and then you can just click image again.
How long does that take? Yeah.
So it takes about half a second to drop down, and then however fast you can pipette, and then the same rate on the way back.
Okay, so less than a second. Okay.
Yeah, less than a second. It can be really, really quick. So the bath actually has like different set positions. So we have a position called escape for cleaning. That takes like, a few seconds to get to, but it is a gap then quite a bit bigger than even what you could raise the old objective assembly to.
Yeah.
But for this load position, it's not much room. It's maybe, I don't know, 5 meters of a gap, but enough to fit a pipette, enough to fit the sample holder in.
Yeah.
All because you don't want to let all the heat of the water out, right? It's all a chamber.
Excellent.
Thank you.
Yeah, super cool.
All right, I see a question from Claire about uncleared tissue sections and how deep you can effectively image into them. So I'd say in Z, the best I've done on a thick slice is about 50 microns. One of the groups, that same group actually at St. Jude that's doing the SimSem stuff, they pretty often image live brain slices on their lattice, and they use about 40 microns at the top. It's what they're pretty happy with for the neuron groups that they're looking at. And they are doing that with either the 50 or the 75 micron sheet that we offer. So you had to do a little bit of trig to figure out what that actual distance is, and that's where about 50 is. We've made longer sheets. We just don't traditionally install them because most people on this system haven't wanted to go deeper than that, just because it scatters so much. It's actually really happy that you asked that, Claire, because it's not like how deep can the beam go? It's how deep can it go and still see something? And that's hard. That is difficult with anything relatively thick and scattering. Now, where we have had luck going deeper is, I know there's groups that have done expanded tissue on the lattice that, it's basically cleared at that point. So sure, you can go as long as the sheet will go. And we've done up to maybe 150 microns on some of our designs. But that's not like the standard kind of sheet that we would do for everyday users.
Thank you.
Thanks, Claire. And then one last thing that someone pinged me about was talking about keeping the beam in focus over time. So something I glossed over in the software side of things is keeping the sheet in focus over a long period of time can be a struggle. So on any multi-objective system, the objectives don't drift at the exact same time, the exact same manner. It's a lovely problem with a lot of multi-objective light sheets that we solved by integrating like a light sheet autofocus. So I'm sure many of you are familiar with things like definite focus and perfect focus on a inverted system, where you're just focusing on the glass. Well, on the light sheet, what matters instead of where the sample is actually where the excitation beam is. So what we do during our long-term captures, we stop the beam from moving, so you actually see the stripes. Run a small piezo volume through that, and then look for the max difference of intensity of the patterns, and then shift the piezo to that new location and resume imaging. So you're then back in focus over time, because if you're doing a 24 or 48 hour experiment, I doubt the room is stable enough to actually keep the beam perfectly in focus over that time. So this is a great way of doing some nice autofocus. Thank you. And I think I don't see any more questions, so I'm going to switch over to just kind of giving you a really brief overview of Slidebook. I don't want to take up past our allotted time, really.
Can I ask one more quick question
of
before
course,
you
go
move
ahead.
on? I was just wondering what is still like the biggest challenge that your users face using the system?
Yeah, I mean, it's kind of twofold, but it was just the scariness that the system kind of looks like. It takes a lot for people to get over. I do understand that. Again, I am a biologist, I'm not a physicist. So when I approached this system the first time, I was like, it's not a microscope. I'll never learn how to use this, right? Like you just think it's scary. So a challenge was both for us and the users was making it easy. And some of that was on us with training and some engineering changes. And some of that was just on users trying it and realizing, oh yeah, actually all this motorization means that like, yeah, okay, I have to mount my sample on a cover slip and then put it on. But okay, if I'm used to using like a different upright light sheet, I had put my sample in a tube before. It's different, but it's not like any more difficult. We're all smart people on the system. But continuing along that path is why we did the fiber coupling of the laser launch, because that just makes it even easier, right? Like there is no alignment besides changing the temperature of the objectives where you might want to check the dye alignment. Past that, you are not looking at the beam all the time. We've actually removed the back aperture camera from the system because we don't need it anymore. We use it on install and then it is never there again. So why make users buy it? So it is both a perception and an actual ease of use. That was kind of the biggest hurdle for the users that I think we've done a pretty good job at solving. I'm probably the most biased person you could ask that because I'm going to say it's super easy. I use it all the time. But I mean, we take it to courses and stuff now. And people have over the years have seen home built systems, seen our earlier versions, and then seen this version. And at least to my face, tell me that they're happy with the improvements. So I have to, I think, believe them. But I do think it's a lot easier.
So it was just the fiber coupling that's what made the difference.
It really made a big difference. Yeah, I mean so that the launch to the path, to the input path was what, like a meter and a half?
Something like that. Yes. Yeah.
And that's a lot of just potential thermal drift. So we actively heat that large launch so it drifts less. Still not perfect. You still get temperature swings overnight. Even the room I'm in now, which with nice HVAC, still drifts a degree or two throughout the day, depending on who's in there. And you'll see the beam drift a little bit. And since the lattice pattern is so pivotal, that was what killed the use. So now in a laser launch with a fiber, any drift in there isn't changing the output position of the fiber. You will change power a little bit over time. So we do still recommend letting the system warm up for 5-10 minutes, but you're no longer turning it on and then waiting for a beam camera to look like a lattice pattern again, where, it now just holds that. And what's really nice is like over the holiday break, I just turn the system off. I turn it back on after the break and it's exactly where I left it. So it was a little surprising to us that it was just that much of the input path, but all of the optics in the breadboard really do hold their position really well. And then obviously if you need to, you can still make minor tweaks. We make annual maintenance visits to all these systems. We'll adjust it then. I mean, it's no different than like, again, a spinning disc. It's going to drift a little bit over a year. This will too, but it is no longer the, oh God, every day.
Right.
Like I don't do a daily alignment. I put dye in here if I turn the incubator off and I want to image something at room temp. But if I just leave the incubation system on, I don't even look at dye. I just image away.
Excellent.
And Brandon, one other comment on that is there was in the early phases of our implementation, we changed every mount that Eric had. We changed some fundamental angles so that they were right angles instead of some interesting Betzig angles. So those early changes also really mattered. So we're talking about increasing stability. There was these periodic changes in which, well, okay, now we're a whole lot more stable than we were before. So the breadboard that we changed that became far more stable so that everybody could do sim and not just the special ones that had the magic room that we found was like, oh, the magic people could, but the others, we traced that to that breadboard. Then we also traced a lot of some of the alignment complications around to the mounts. So they're all now all Polaris mounts, like the changes of the mounts that are there was just a really different versus, hey, what do we have in the lab that's then became perpetuated through eternity in the Betsy designs, in the home build designs, versus what would we do if we were engineers designing this from scratch?
Right.
How would we make those same angles? How would we propagate the sheet through the system like the design had it optically and the uniqueness of the design, but how would we make that fluidly easy for everybody to use? So we made these interim changes that were phenomenal, but then this one was like, okay, now anybody can use it. This is nothing at that point.
Awesome. That's
Pardon my interruption, Andrew. I had one more thought to add into the mix here today. By the way, Sam, director of sales at 3i. I'll turn my mic off now.
All right, so I'm going to switch over to some fun slidebook stuff. So let me share this now. And hopefully you are seeing, there you go, my screen.
Yep.
And what we have here is unhealthy cells. I will apologize for the quality of the samples I have here today. I'm also a resident biologist, which just makes it extra embarrassing. But the samples are just not happy right now. I don't know what this batch of cells is doing. I grew them up a week ago, and they are just not recovering nicely. So excuse that it's slightly rounded up, but it's still fluorescent. We're still gonna at least look at it and give you a nice overview of the software. So if I just like go live to give you an idea, and that's just the fluorescence 2D image that I'm sure many of us are used to looking at a lattice. Be it home built, be it ours, be it the Zeiss one, you're always looking at the sample at a slight angle when it comes to looking at through the light sheet camera. But something that was really nice that we've added to it is just even a bright field LED can make finding samples pretty easy. So this is just gives you this pseudo DIC effect on the camera. So you can see as I scroll through here, this is oblique illumination because we're actually illuminating through the excitation objective, but we're detecting through the standard light sheet cameras. So it gives it this relief style image that you'd get from DIC, but it's not actually DIC. For cells, this is nice. I mean, this tells me as a somewhat trained biologist that cell is not happy just by looking at it. But what this excels at is large samples. So even the C. elegans I was talking about before, we always still found them via transmitted light. They're huge, right? So you just immediately see a shadow before you even get to the worm itself. But when I've done Drosophila embryos on here, when I did a Arabidopsis on here, this transmitted light was how I found everything. It was just faster than saying, okay, well, I think I'm going to find fluorescence in this section of it. No, I'm just going to look for the whole blob of an organism. So that's really nice. But one of the things that we've really tried to make easy to use on the system is just doing captures and looking at the data quickly, doing processing quickly. So there's a really quick overview of imagining a basic capture that we'd set up. If I just turn on the 3D settings, I'm just going to capture a moderate volume of this system, and it's incredibly bright. This is LISO bright red. My counts on these flash 4s are incredibly high, so I can even drop my exposure time down quite a bit. And I'm only using 10 milliwatts out of the laser launch to show you how bright this is, but also show you that you don't need a lot of power for moderate samples too. So this is the other reason where we were okay with shifting away from that super high powered launch, is we don't run the lasers that high all the time anyway. So I'm just going to do this not as a time lapse first, but just as a quick 2D image or 3D image. Flies by, right? Where I can now scroll through and we can see what we're looking at here. So again, excuse the less than healthy looking cell. This is supposed to be lysosomes, as I said. They're there. They're just not great looking. But again, even for users that are getting used to this, kind of back to what Claire had asked, was okay, I'm not used to looking at my sample from the side. So what's this actually look like? We made it really easy to just take this data without doing any sort of processing and just look at it in 3D. So our system inherently is doing a digital deskew and then even a cover slip rotation where you can just look at this data now normal to what would be Z to most people, or without that, you can just look at it quickly in 3D and see again, clump of less than happy cell, but some lysosomes are actually there. But doing this right after capture, you don't have to process or do anything to look at it in 3D. I'm sure most of us are familiar with doing something like deskewing. Obviously, that's in our software as well. So if you wanted to take these data and move them elsewhere, Slidebook's deskew is incredibly easy to use and incredibly fast. So you get a bunch of settings that you can select, things like if you want to do that cover slip correction, if you want to make maximum intensity projections right away. But one of the big improvements we made about a year ago was GPU accelerating this. Not that deskew was inherently a difficult or slow process before, but we've made it incredibly fast now with some CUDA-based code, and also can output it to a separate file so you can be accessing that as it's going. So I'm going to just put it in the same file, click apply. You're basically not even going to see a progress bar go by because this is not a large file. Went from 60 to 50 megs. But as an example, if we had, this is just my time lapse that I did before. We're kind of welcoming everyone. This deskew is still really quick, even on time lapse data. So 2 channel data point, 20 time points, and this is making about 6 gigs of data, and it takes about 5 seconds. So still really quick and easy to use. There's our 5 gig output file. Meant to be, you know, now I'm taking my data that I've rotated it and I put it elsewhere in the software or in other software packages. The other cool stuff that we've done, I mentioned the autofocus before. So there's a nice little autofocus setup. We can set up how frequently it does that. Our software gives you really easy multi-point control. So if we have our sample here, I can clear this. I can set that as a point, just like you do on many systems. And there's not a lot of cells in this cover slip, so I'm just going to go to like a random point and set another point. And then if you ever want to go back to it, you can double click visit. You can do this obviously as scripted captures where you'll, mitotic cells are the classic example of this, right? Like you don't always know inherently which cell might go into mitosis at the same moment if you haven't done some sort of synchronization. So you might have 30 fields of view mapped. So these cells are good candidates, and then you image those overnight. And instead of doing one over and over again, you can get some nice repetition out of that. I see a chat question come in. So I'm just going to take a quick look at that. Someone asked about cover slip correction. I guess I should explain that a little bit more in detail. So cover slip correction is a term for rotating the sample so that Z is now normal to like how we see Z on the sample, where Z is now up and down relative to the cover slip. When you collect data on the lattice, your Z to the detection objective is at an angle. So sure, you could just spin the data 7 or 57 degrees. But if you want it to be up and down as you just scroll through in Z, that's what cover slip correction is. It's not actually imaging through the cover slip. So thank you. And thank you for Vanessa too. I realize I'm running out of time. So I'll actually stop and see if there's anything people want to see in the software before I just eat up too much of your time rambling.
For your output, for your data output, what format do you guys typically write it out to?
Cool, great questions, Brandon. So we are a commercial entity. Our default software package outputs what we call slide files. It's a proprietary format. We've worked with bioformats. You can open them in Fiji and other rendering packages like Amaris and Erebus and stuff like that. But we also now have a new file format that is Python based. That just slide with a Y at the end, slidey. And that is NumPy and YAML arrays as a file. So you can actually be reading that and doing something with it as you're acquiring. I saw Zeiss talk about extensively how they've been working on that as like external processing code. We're doing it in a slightly different approach where there are a lot of third-party solutions, not necessarily commercial, but written by labs that are really helpful. I know Tally Lambert has worked on this extensively and other groups where they have all these packages, most of them Python-based, that you can just run these scripts on our data now without having to do any sort of file conversion. You can just use it as is with that new file format. But it just depends. We give users the option which one they want, because some people like the old one where they have one file where everything is a single thing and not a folder of a bunch of what they call gibberish if you're not used to Python. Sure. But yeah, thank you for asking. Anything else I can show? Or I have 1 minute left technically on my allotted time, so I will I will not keep anyone late.
I just have one final question because the first time kind of seeing, is the analysis, can you do that in Slidebook as well? Is pretty much you just acquire and do everything in Slidebook to the end? Is that?
Yeah, really can. So this is where I could give hours on this.
Sure.
But sure, like even any of this, you can deconvolve it in Slidebook. So we have our own decon package back to the 90s. We've modified it since then, of course, but it's still core to what we've done. So we have, I just kind of open up a file. You can see kind of the interface here, where you get to use different point spread functions that you've acquired on the Lattice. I'm sure many of you are used to looking at beads. I look at beads in the system all the time, so you can take those point spread functions and import them and do that in here. We have other analysis like particle tracking, different segmentation for quantitation of data. If you're doing FRAP, we have a really nice FRAP analysis that gives you the different coefficients that you want, looking at the bleaching and the binding. But yeah, all this within Slidebook. Then you can export your data either using our 3D viewer. We've done some storyboarding, much like you would in all the big rendering packages nowadays, where you get keyframe scripting of everything. Or you can take your data elsewhere. I know obviously Fiji is here for a while. Nepari has become very big in the Lightsheet community when looking at data, but also the commercial packages, Arevis, Amaris, Avia, Amira. They all sound similar, which is what I love. But that's fine too. And most of those can open slide files because we've worked with them for years too. With that, I think my time is up. So I'll just say thank you one more time. I'll stop sharing. And I provided my email. Obviously, any of you can reach out with questions. I am at workshops all around, so please come see me. I know Bina has some involvement in the Lightsheet workshop that'll be in Woods Hole in May. So this will be there. So if you're going to be there, come and see me and the system. But thank you again.